Hi, welcome back to Discovering Patterns in the Microbiome. In this video, we will talk about beta diversity again, specifically the UniFrac distance metric. So UniFrac is one of the many metrics that's implemented in QIIME. We're going to be using the Guerrero-Negro microbial mat data set that we discussed in the beta diversity video. And previously, we had looked at three different beta diversity metrics: Euclidean, chi-square, and Bray-Curtis. And we saw that they all worked reasonably well in these data, but chi-square perfectly recovered the gradient in the data, the gradient of the depth of the sample in the microbial mat. I didn't mention there's this U shape there. That's a very common phenomenon here, and we're actually gonna address that also later in the class. So these metrics work pretty well in most cases, but one thing that they're not taking into account is phylogenetics. So they don't take into account how closely related each of the species is to every other species. So for example, if I have a bunch of species in my gut and you have a bunch of species in your gut that are totally different species, but they're very closely related to mine in terms of evolution, then we don't want to say that our microbiomes are completely distinct. We want to have a more sensitive test where the test would recognize that our microbiomes are different but more closely related, say, than to another person who also had different bugs, but they were very distantly related to ours. So the phylogenetic distance metric is called UniFrac, and it's using a similar approach to the phylogenetic alpha diversity that we learned about. So please recall in phylogenetic alpha diversity, we were basically counting up the fraction of the tree that is covered by a particular sample. So if you look at the phylogenetic diversity of the red sample, it covers a lot more of the tree than the blue sample. So it would have a more phylogenetically diverse set of species than blue, even though it might have the same number or similar number of species. So we're going to use something very similar for beta diversity. And what we're doing is taking the fraction of the tree that is unique to one sample or the other, according to its species. Here are three different examples. So the first one, if you have identical communities, this is where basically each community has bugs in the exact same places in the tree of life, in which case none of the tree is unique to one sample or the other. So the UniFrac distance or unique fraction of the tree would be 0. You could have somewhat related communities, such as the middle figure here, and this is where they share a lot of their evolutionary history, say at the genus level on up to the phylum level, but when you get down to species or OTU level, they tend to diverge. So in this case, maybe about half of the phylogenetic tree is unique to the red sample or the blue sample, so they would have a UniFrac distance of 0.5. Then finally, we have totally unrelated communities. So this is where the bugs in the red community are completely unrelated from a completely different lineage from all of the bugs in the blue community. So the whole evolutionary tree is unique to one sample or the other, and they would have a UniFrac distance of 1. So this is UniFrac. It's the percent of the observed branch length that's unique to either sample. And there's also a version of this called weighted UniFrac that takes into account the relative abundances. It's no longer just the raw unique fractions. Now the branch lengths are weighted by how much there is of each bug in each sample. UniFrac is just one of the many distance metrics that's in QIIME. And so we'll use it typically as part of this approach where you start with the full tree that contains all of the bugs from all the samples. You use it to calculate the UniFrac distance between every pair of samples in your data set. That gives you a distance matrix, which is shown here. Then you can do lots of things with the distance matrix, but the most common one is the principal coordinates analysis that we talked about that will then allow you to visualize those distances in 2 or 3 dimensions. Now, I mentioned weighted UniFrac uses the abundances of the bugs. So because of this, weighted UniFrac tends to place more emphasis on the dominant bugs. It's not going to be influenced much if you have some unique bugs that are really low abundance. Unweighted UniFrac, on the other hand, because it's only using presence and absence, it often emphasizes the rare bugs. So let's think about this. If bugs are really common, in other words, they tend to show up in a lot of people, then they'll be present in a lot of people. And so they're basically not going to produce any unique weight on those branches. Whereas in unweighted UniFrac, in unweighted UniFrac, whereas if you have minor bugs that are absent from a lot of people, then the mere presence of the bug in a particular person is going to add a lot of weight along its branch. So that's kind of the way I think of these 2 different distance metrics. Weighted UniFrac emphasizes more dominant bugs. Unweighted only considers the species overlap between the samples. So let's look at how you would actually make UniFrac distances. So we'll go back to the same code document from the previous video. This is the beta diversity document. And we'll skip down to the UniFrac section. So if you want to calculate UniFrac distances in QIIME, you can use this. You can do this using the core diversity analyses script, or if you want to do just beta diversity, you would use this command, beta diversity.py. You input an O2U table, an output folder, and because we're calculating UniFrac distance, you have to pass in a phylogenetic tree. So there is a phylogenetic tree included in the course repo. If you go into the data folder, it's under ref green genes, and you see there's the reference phylogenetic tree that's used for closed reference O2U picking. So you would run this command on whatever data set, assuming you had an O2U table, on the command line, not in R. Now let's go back over to R and load in those UniFrac distances. So we use QIIME to generate the distance matrix, then we're going to load it into R to do some visualization. So here's the command to read in those 2 tables. We need to make sure that the distance matrices have the same sample IDs as the mapping file and the other distance matrices we've calculated, and this has to be the same samples in the same order. Then we'll calculate the first 2 dimensions of the principal coordinates in the same fashion as we did with the other distance metrics. Now let's plot unweighted UniFrac. So here's what we get for the sample. Looks pretty good, actually. Basically comparable to chi-square in terms of recovering that gradient on the first axis. Let's look at weighted UniFrac. Not bad, but it's collapsing one of the ends of the distribution. So it looks here like the unweighted does better at telling the difference between different layers in the microbial mat. So these are the 2 UniFrac distances. I wanted to make a quick note. If you ever want to actually generate a PDF version of one of these figures, I put a little code in here to help you do that. It's the same plotting command. You just call PDF beforehand and dev.off afterward. So now that we've generated these 5 distance metrics, I thought it would be interesting to ask a few questions about how they compare to one another and which one is best. So first, we can plot the pairwise scatter plots of the 5 different distance metrics. So we now have Euclidean, chi-square, Bray-Curtis, unweighted UniFrac, weighted UniFrac. So the way we've done this is we turn each of the distance metrics into a numeric vector, and then we use cbind. This just binds them as columns into a big matrix. I'm going to borrow the names of this matrix from I'm just going to add these names in here. And then there's this command pairs, which makes this nice little scatter plot. So what can we see here? Let's look. This catches my eye in the lower right. Looks like unweighted UniFrac and weighted UniFrac are actually quite close to each other in terms of the overall distance matrix in this data set. Chi-square and Bray-Curtis are fairly similar. Chi-square and unweighted UniFrac are kind of similar, but there's a bit of difference in these points here. Looks like unweighted UniFrac, looks like the two UniFrac ones and Bray-Curtis are highly correlated. And Euclidean's a bit of an outlier. It tends to be different from all the rest of them. So that's just a nice way of comparing a whole bunch of variables to each other. We can also plot the Pearson correlation or display the Pearson correlation for each of the pairs. So we just use the core command on the same matrix. And here's what we get. So looks like Bray-Curtis has 93% correlation with unweighted UniFrac, 94% correlation with weighted UniFrac, and so on. The last question I thought would be interesting is to ask which distance metric did the best job of recovering the physical sample distances based on the depth of the layer. So in the metadata file, there's one column. You can open it in Excel. There's one column that gives you the layer, just the number, like the first, second, third, and so on. There are 10 layers. There's another column that gives you the ending depth of that layer. In other words, how far down did that layer go? And so that's what we're going to use to evaluate the distance metrics. So the first thing we're doing here is kind of clever. We're calling dist on the end depth of the samples. So dist is the same command we used earlier for calculating Euclidean distance. And what this is doing is just saying how far apart are samples in the actual mat. How physically far apart are they, like in centimeters? And for that, you would use Euclidean distance. That's what Euclidean distance is for. It's measuring like physical or geometric distances. So, so we're calculating that distance matrix, and then we just convert it into like we stretch it out into a single long vector. Now, I'm going to make a list of the other 5 distance metrics. I'm going to reuse the names from our distance matrix before to name those the elements of that list. And then I'm just going to loop through the list or walk down the list. And for every distance metric in the list, I will convert it, basically stretch it out into a long vector, just like I did the physical Euclidean distances. And then I'm going to run a correlation test of the physical distances. I'm sorry, these are the ecological distances that I just stored in the variable d with the physical distances that I stored in the variable y up here. I will use Spearman correlation. Spearman correlation doesn't take into account the magnitude of the values, but only the rank. So if the last sample is like 50 centimeters away from the previous layer, and that layer is only 1 centimeter from the other one, Spearman's not going to look at how large those are. It just takes into account the rank. It's a non-parametric test for correlation. So let's find out how correlated each distance metric is with the true distance, physical distance in the mat between samples. Euclidean. Looks like we have a correlation of 0.385. That's highly significant. Here's the p-value. So it's not bad. But now let's look at Bray-Curtis. 60 looks like 62.7% correlation. Chi-square, we're up to 0.75 correlation. Now let's go back to the UniFrac. We have 0.53 and 0.43. And so what you see here is that the chi-square actually does the best job of recovering the overall distances, even though it doesn't take into account the phylogenetic relatedness of the samples. Now, so I wanted to mention one neat trick that you can use in beta diversity. So far what we've done is we take the overall beta diversity and we collapse it down to 1 or 2 or 3 dimensions using principal coordinates analysis. Another thing you can do that can often be quite useful is if you have a reference group of samples, say your control samples or baseline samples of some type, for every other sample in your data set, you calculate the average distance of that sample to all of the reference samples. And now you have one value for each sample. So you've gone from this giant distance matrix to a single set of values for all of those samples. And that was a trick that was used to make this somewhat well-known figure 1 in the global gut study that we've talked about a few times. So the trick here was you plot each child's average distance to all of the adults in their country. So now beta diversity is this single variable that's shown on the y-axis here. It's UniFrac distance between children, or a single child, and all of the adults. And then you can plot that against age. So as you plot that against age, you see kids start off very different from adults, and then they become more similar to adults, more similar, until they kind of level out at age 3. And that was a very nice way of doing that analysis and visualizing it. So in summary, beta diversity measures diversity between communities, as we've said. UniFrac, which is phylogenetic beta diversity, is a very useful approach, very commonly used. Most people use UniFrac and sometimes will add in Bray-Curtis. However, chi-square is often best for gradients. And although it's not phylogenetically informed, so maybe there's some kind of happy medium between the chi-square approach and the UniFrac approach that would be even better at recovering gradients. And finally, I showed you a little trick where you can convert beta diversity to a single variable by a single valued variable by comparing each sample's average distance to some kind of control or reference group.